Well, thank you very much then for being here. It's a pleasure then to start this second Turing lecture. It's a great opportunity to thank again all the organizers and you for being here and sharing your research, your experiences. It's been a very nice experience in this discussion meeting. So let me start now this second part of the lectures with a more thorough description of the biomechanics that I was talking about. So in the first lecture, we covered the question of birdsong learning at the level of behavior. And now I'm going to be talking about the periphery. And then the idea is that tomorrow we'll go a little bit towards the brain. So that will be the path from the environment through the biophysics to the brain. But I also wanted to talk a little bit, especially having in mind that this was a school or that there were going to be students here. I wanted to discuss a little bit the complementary approaches of interpretable science and data science in a research program. So what I did is I adjusted a little bit the story I wanted to share, stressing the differences between these two approaches and the kind of lights and shadows of each of these two approaches to a given problem. So what I was talking about, and the subject I've been working for 25 years, can be summarized in like 10 seconds in this movie that displays, as we were discussing, the vocal organ that in the case of ossine birds is a bipartite structure where there are two pairs of labia which eventually, because of the action of some muscles, get together, and when the air comes through, modulate the airflow. So the time derivative of the rate of mass injection of mass at the base of the trachea generates the perturbations of density that become sound. And the bird has control of these two sides with a set of muscles that I'm going to be talking about. So the program that I followed when I approached this field coming from nonlinear dynamics was the typical interpretable science approach, which consists in the following: having a phenomenon that in this case was a song, which for me it was a surprise, was a complex sound like this. And by complex sound, I mean that in this species you have frequency modulations and harmonic contents that are not trivial to anticipate, and that give this particular sound. And my purpose was to try to find whether it was possible to find a low-dimensional dynamical system which could reproduce that phenomenon. And as I was describing in the first lecture, what I tried to do is to describe the dynamics of these labia, okay? Write differential equations for those, and then simulate the pressure waves and generate the synthetic sound. The hope was that with a low-dimensional dynamical system, I could do that. And of course, I would have a few variables which I could interpret. I couldn't measure them, because at the time I couldn't measure them, but I could interpret them. And in terms of those variables, I was going to write equations. How would I write equations? Well, in a very simple matter, these equations would be Newton's law applied to the midpoint position of the labia. So the basic physical intuition that I had at the time, it was that, like in the case of the human voice production system, you would have vocal folds with two modes that would be active. One would be a lateral mode, the other one would be a wave-like mode. And if they are active at a precise phase, what you would have is that you would have an average pressure which is larger when the labia are moving apart, and a lower pressure when they are moving together. Why? Because if you have a large airflow, the average pressure between the labia when you have a conversion profile will be similar to the pressure in the airflow, in the air sacs. It's going to be high. And then when you will be approaching with a diversion profile, the average pressure will be similar to the atmospheric pressure will be low. So therefore, you would have a force in the direction of the velocity and therefore a way of impressing and transferring energy towards the labia. So that was the basic idea. With that kinematics in mind, what I computed was the pressure in between these two labia, and those are just Newton's equations where I take into account a restitution term because of the elasticity of the labia, a nonlinear dissipation term because when the labia approach the trachea that is containing them or they approach each other, they will dissipate very rapidly the energy. So you have a nonlinear dissipation. And eventually here you would have the nonlinearities that come when you do the Taylor expansion of this pressure, which you compute by doing basic thermodynamics. No? So when you do that, in the framework of interpretable classical system, what you try to do is now generate the sound. How do you generate the sound? Well, y is basically the derivative of x, that is the time derivative of the area, okay, here in between this valve. And as I was saying, the time derivative of the airflow basically is the sound. I don't know if I'm being clear. Let me write it down. This I can do with a simple equation. You have that the flow is going to be the density times the average velocity of the air in between the labia times the area. The area is the variable x times the transversal length. So if you take the derivative of this is very slow, so basically it's the derivative of the area. That's the reason why y is the variable that is important here. The velocity, the air velocity, is a slow variable that goes in the scale of the 100 milliseconds, 200 milliseconds, and we're describing a very rapid motion in the order of the kilohertz. Okay, so now we can generate the sound if we know what the bird is doing with this parameter here and basically this parameter here. Why? Because when you want to sing, you want to have a large expiratory pulse, and that is the way in which you make the labia start to oscillate. And while you are doing this, basically this expiratory gesture, you can change the restitution constant of the labia because you can stretch this labia, okay? And that's the way in which you can control the acoustic features of the sound. And if you come from nonlinear dynamics, like it was my case, then that model, even if it has only two variables, is complicated, is too complicated. And you want to do what in the field of nonlinear dynamics is to go to the normal forms. Whenever I talk to a younger person that wants to study dynamics, I say, just spend a couple of months studying normal forms because your life becomes incredibly easier. Because if you have a problem like this one that is full of parameters, there is a systematic way of doing nonlinear changes of variables that take away every term that can be taken away. Only the resonant terms will remain, so your problem will have just a few parameters. So your life, after doing that homework, will be incredibly easier because you will have to move less parameters if you do a numerical integration. So if you are not lazy and you take a couple of days to do the hard exercise, then your life becomes much easier. And I bet you that there is a Russian mathematician from the beginning of the 20th century that solved this problem already. You have the picture done and everything. So you save a lot of time by doing normal form analysis. And in this case, I'm showing you exactly what happens in the parameter space of this problem. And I did this, well, Anna did this painstakingly, no? It was very hard to do. She had to do all the bifurcation diagram of this particular system, okay? And you have all these qualitative different regimes where, for example, here the pressure, here you have the pressure, and here you have the tension of the labia. So for low pressures, the labia are quiet. If the pressures are high, they are going to be oscillating. But the way in which you lose the stability will be different. Here is what we call a Hopf bifurcation, which is a way in which oscillations are smoothly being born. Here what you have is what is called the saddle node in a limit cycle bifurcation, where oscillations start in an explosive way and stuff. Now, if you, instead of working with this, you work with the normal form, what you get is exactly the same set of dynamical regimes. That's the advantage and the beauty of the normal forms. You know that this very simple system, that where you have eliminated every possible term, will have the same dynamics that the original one. So for a dynamicist, this is a dream. This was my dream. A complex problem, let's see if I can model it with something that has only 2 parameters. And also it's nice because I thought, well, if I have to measure 25 parameters, I will never set up a lab. When I thought, well, maybe I have to measure 2 parameters, maybe I can set up a lab because I have to learn 2 things to do, right? So that was the idea behind doing this normal form analysis. So as I was telling you before, you can then start an oscillation by doing this kind of path in parameter space, and you will start oscillations which are very smooth because in a Hopf bifurcation, oscillations are being born in a smooth way. And if you have a path like this one in parameter space, you are going to be starting the oscillations because of the collapse of a saddle and a node, and therefore you are going to have a system that is slowly staying here and then rapidly moving around, so you are going to have oscillations of a very high spectral content. But basically, the idea is that, for example, if I want to generate a frequency, a syllable that has an upsweep behavior, what you have to do is turn the oscillations on at the region where the frequencies are low, and then start to increase the tension of the labia until you turn the oscillations off. So therefore, you're having syllables that go up. Okay? So it's only the phase difference between 2 simple gestures that you have to control to have a very sophisticated acoustic feature like the frequency modulation. So in the case that I was telling you before, you will have a canary syllable like this, and this would be the model that I was describing before. So after this first work, I decided to set up a program, which is what I've been doing during the last years, and I'm going to describe it, and then I'm going to list it, and then I'm going to describe it. So I had to set up a lab to measure these parameters and do some animal physiology, to measure syringeal muscle activity, which are the ones that are going to be controlling these tensions, because when you have the trachea, you have the labia that are inside between 2 cartilaginous annuli, and the muscles inserting here. So when you contract the muscle, you increase the distance between these annuli, and the labia that are attached there get more tense. So increasing the muscle activity increases the tension, and therefore the restitution constant of the material. So I had to develop some phonation models, generate synthetic songs, which could be informed by physiological gestures. Once that I have a model, if I take the data, I have to put it into the model and see what happens. If the model is okay, I should get the right sounds. So I wanted to find the physics of phonation for different avian orders, because as I was telling you, Osine birds have these kind of seedings, but there are other orders of birds that have other seedings and have other physics involved, and they will have different sounds with different timbres. I had to develop a respiratory model because I had to measure, I could measure the air sac pressure, I could measure the activity of the muscles, but the actual pressure that comes out of the system is also obeying another biomechanics, which is the biomechanics of respiration. And then measure neuronal activity during gesture generation. That is something where Anna got involved, as she was mentioning. And one of the interesting aspects of it is that it was a very nice way of testing whether the animals were believing that this was good enough or not, measuring the response of selective neurons to real and synthetic songs. Then we went into the wild to see what happens with the wild animals, trained juveniles with synthetic songs, and use models to interpret avian dreams, which is something I'm going to be talking about tomorrow. This is something that I would love to do, but I'm too far away and maybe too old to do it, to try to find a mean field model in order to try to link these very sparse measurements that we get of neural activity with something that can be compared macroscopically with these physiological gestures. This would be like the ultimate dream of this program. But the first thing started with this idea. I couldn't go and look at the labia, but what I suspected was that I was going to have basically a relaxation oscillator with a couple of modes. So when you look at the dynamics of this, in principle, you would get the dynamics of relaxation oscillator. Let me actually do a little bit of algebra here, and you can convert that system, if you develop the things up to third order, to a very old and classic van der Pol oscillator, which if you take a nonlinear dynamics course, it's going to be like one of the first exercises that you have. You will have a couple of equations in here. A null Klein, that means the equation where this is equal to 0. Another null Klein, that is where this is equal to 0. So the intersection of these two is the unique fixed point. And then what this system is displaying is a dynamics where you jump very fast here, dissipate the energy, jump here, dissipate the energy, jump to here, dissipate energy, okay? And do this kind of labial motion. This was all a conjecture, and I couldn't measure this for a long, long time. But a few years ago, maybe 4 or 5 years ago, Koen Elemans from Denmark, a good friend, made an ex vivo preparation of a pigeon seedings, and he got movies like this. This is the first time that I could actually see what was going on with the labia. And this is one beautiful example for me where my prejudice that, doing interpretable science is great got into conflict with the temptation of using artificial intelligence, because with this movie, I knew I had all the data, but how to get it, right? Because what I said is that my equations would be describing a midpoint position of the labia, some point in here. How am I going to select it? How am I going to track it? So fortunately, by the time that Koen could make this movie, neural networks were fashionable. And I decided that I would try to lower the dimensionality of the problem with an autoencoder.
Can you see the fast mode and the slow mode there?
Let's go there, because that is the question, exactly. That is the question. Of that movie, I could get a lot of information, but that is exactly a question that I could compare with something that it was important to me, that there was a difference of scales of some sort. So let's see if with this artificial intelligence baby, with a simple autoencoder, we can figure this out. So, are you familiar with this beast? And what about the younger people? Who is not familiar with an autoencoder or a neural network? Okay, right. So, this is a neural network where every one of these units is a very simple unit that will process the information in this way. You will have an input, and you will apply a nonlinear transformation here. Could be a sigmoidal function, could be a ReLU function, some nonlinear function. So whenever you have to compute which is going to be the value of this variable, you are going to be computing which is the value of the units in the layer before. You are going to add them together and you are going to apply the nonlinear transformation that corresponds. This is the rule. Now, what is the input of this beast? The input of this beast is the information that you want to process. In my case, for example, the frames of my movie. Each frame is a matrix of values. Each point in the image, okay, is going to have a couple of indexes, I and j, and it's going to have a numerical value, which is a level of gray, between 0 and 256. So I can put all that information in a vector, and that is going to be my input. It's going to be processed according to the levels of the weights of these connectivities. So there are going to be numbers here that tell you, if I have a 1 here, now the input that corresponds here is going to be weighted by some number here. So at the beginning, it's going to be a bunch of random numbers. And what I want this network to do is to copy whatever I have here in here. So it's the most silly thing you can imagine. You can say, well, if you want this to be equal to this, why not to write the identity? You could. But the challenge here is precisely to ask the network to have a minimal number of units in what is called the latent space. So you compress information like a bottleneck. And you want your set of frames to be mapped 1 to 1. That means each frame in the movie is going to be an input, and you want this to be copied identically. The next frame, you are going to put it here, and you want to copy it, recover it here. So the thing is that if you compress it too much, you won't be able to do it. Because if you compress it too much, maybe 2 inputs will go to a unique point, and then you can recover it, okay? So if you compress it the right amount, you can have that every point in your, every object in your input is exactly copied, and therefore here you have a 1-to-1 representation of the states. So it's a wonderful way of doing a nonlinear compression of your information, where if you have a dynamical system, every point in your movie is mapped into 1 point in this low-dimensional space. And since every point has a unique state associated to it, you can call it a phase space. So with an autoencoder, you have an automatic way of getting a phase space of your dynamics. That's the idea. So now I can process the information, as I was telling you, by asking frame by frame to reproduce whatever I have in my input. This is what I call the latent space, where I'm going to be encoding the data. And I'm going to be representing this, I don't know, 500 by 700 pixels into just 2 variables. Now, I didn't pick them. I didn't say, well, the important thing is, I don't know, the midpoint position of one particular point. I'm asking this neural network to make a compression of the information and unveil the phase space automatically. So if you do that, and again, maybe 2 is too small, maybe you need 3 variables, maybe you need 4. It depends on the dimensionality and the complexity of your data, of course. But if you manage to have a very low dimensional representation, then you're done. You can call that a phase space and recover the equations from the projection there. So a good encoding is a good encoding where there are no self-intersections of the data. Well, this is the typical diagram that you will see when someone is doing some sort of neural network analysis. The description of your neural network, in this case, is this autoencoder. This part is what is called an encoder, and this is called the decoder. It's a symmetric image of the encoder. Typically, you have, which is the number of entries and the number of units in the different layers in the middle. Then you have the latent space. As I was telling you before, here you have a symmetric reproduction of the stuff. And in here, what you display typically is the mean square error as a function of the epoch of the training. What do I mean by this? Mean square error is just to take the difference between the image and whatever you get out. So at the beginning, you have a collection of random number. You put an input, and you get whatever. Noise. The nice thing that Geoffrey Hinton did in 1986, and although it was not explicitly said, is probably what got him the Nobel Prize last year, is a method called backpropagation that, given the difference at certain stage between what you needed and what you are getting, how to change in a training iteration, how to change the parameters so that in the next trial, the difference between this and what you get here is smaller. So typically what you do is you display in this set of iterations, which is the successive errors that you are getting. So this is the typical diagram that you see, okay? Now, why do I have two colors? Because I have a training set and a test set. Well, we were talking about that the other day, right? Who was I talking about? Training and, it was with you, yeah. So whenever you have data, you have to keep a little piece apart, which is the data that you're going to use to test the data, and some data you're going to use it to train your data. So that's the reason you have to. In this case, there is no overfitting. That means that not only you can fit a good function to your problem, but also it can generalize a little bit. And in here, what I'm showing you is which is the mean square error as a function of the latent space dimension. As I was telling you before, maybe you have that 4 dimensions is great, 3 dimensions is great, 2 dimensions is great, and if you want to reduce it further, then it doesn't work. So in this way, you can see which is the good dimensionality of your phase space. When you stop decreasing, there you are, okay? So from here to here, I gain, okay, because my mean square error is smaller, but then if instead of using 2 dimensions, I use 3, the system doesn't improve. That means that 2, it was okay. So now I have my data, and I know that with a 2-dimensional projection in a latent space, I can reproduce every state in my movie. So now, Nelima's question, do I recover what I wanted? So here it is, the velocity computed from the frames, and the projection in the latent space. And if you notice, here you have a denser set of points and a denser set of points, and a less dense set of points here, and a dense set of points in here. That means that you have 2 slow regions and 2 fast regions. So this was the first time that I thought, well, maybe it was okay to assume that there was a relaxation oscillator in this problem. And I did it without needing to go and make a very specific selection of which point to obtain from that movie. I just processed the movie, sending it just to a neural network with the right question. Because the question is whether what you're looking for, you can get it from here. Now, this opened for me another line of research, theoretical in nature, of whether you could prove that if you process a movie that comes from a dynamical system that has certain topology in phase space, whether really you can prove that in latent space you have the right topology. I'm not going to be talking about this, but you can prove it. So this is really a phase space, and the topology that you get is the right one. So the moral of the tale for me is that I don't have a great sympathy for neural networks and data science, but man, sometimes they allow you to do things that you couldn't dream of doing it in any other way. So how to use them in equilibrium, it's an interesting challenge. Going back to the problem of measuring this, as I was saying, if you work with the normal form, it is clear that you only have to worry about two parameters, the restitution constant, which is the tension, and the pressure. This is what you get from the normal form, alpha and beta in my equations. So these are two things that we could learn to measure. So, yes. Okay, so, oops. So these are the time scales. Let's say that the bird is breathing. The bird is breathing. So these are the fluctuations of expiration and inspiration. And then you want to generate a syllable. So you generate a pulse here, and this is a slow dynamics. This could be 100 milliseconds. Within that window of time, you will have the rapid oscillations of the pressure fluctuations. What I'm analyzing in the movie that we saw before is this very fast dynamics. So for all these 3-4 oscillations that we analyzed, the value of this parameter, the pressure, is basically constant. Okay, so this is a time scale that is much slower than the ones we were analyzing. So I'm talking about different oscillations when I talk about the fluctuations of the pressure and the tension, and when I'm talking about the oscillations of the labia. So the one that we were analyzing before are the very fast oscillations of the labia, which are self-sustained. You don't need to move any muscles to generate them. It's like you blow air in between two pieces of paper and they start to oscillate. So now I'm focusing in the other scale of the problem, which is the slow one. And now this is the one I'm going to be dealing with, the slow one. Okay? Great. So how to measure the pressure? Well, we have this very nice poster. Your poster. So you can check her poster. You insert a cannula in the posterior air sacs and you send it to a Fujikura pressure transducer, typically what we are all using, that goes to the backpack. And then to measure this is for the pressure, and then to measure the tension, what you have to do is you have to insert this super tiny electrode in here, send it under the skin to a preamplifier here in this big backpack, and that's going to record it afterwards. So for me, it took me many years of development of chirurgical techniques, electronics, collaborations with Franz Goller, who is teaching us all these techniques. And the typical data that you get when you do that is this. These are the pressure fluctuations when you're generating song. So this is the song expressed as a spectrogram, that means as a function of time, which is the frequencies. And these are the time series that correspond to the pressure and the muscle activities of different muscles. Now we're talking about the slow motion of the fluctuations, okay? So you have expiratory pulses, and in the case of the zebra finch, what they do is motifs where they repeat the syllables in a very stereotyped way, and every bird learns one song, just like yesterday with the chingola. Well, it's the same with the zebra finch. So whenever it's repeating these 2 times, you have the same syllable that I'm plotting in green, and you have the same gesture in green here, and the same gesture, and the same gesture in pressure, with the variability typical of any biological system, of course. So my question is, now if I take the path in parameter space that is suggested by the data or simple trajectories, and I go around a bifurcation diagram of a normal form, how similar or how different is the synthetic song that I will generate? So I have a copy, which is, this wasn't very smart. I should have put them in the other order. So you have the recorded one, and you have the synthetic one. Let's try to hear how different they are. One is a recorded sound. The other one is the integration of a normal form. That means a differential equation of two equations that have only cubic nonlinearities with only two parameters. So it's really a simple thing. So that to me sounds very similar. Okay. Oops. And as I was telling you before, we did this experiment with Anna when she was in Chicago, where we played these songs to a sleeping zebra finch. And when the bird was exposed to the birdsong song, you have this response, and it's the same response that you got with synthetic song, response that you wouldn't get with the reverse song or the context-specific song. Someone asked me yesterday a question which I would love to do systematically. I think it was you. Whether different species could be placed in the same bifurcation diagram. And I don't have the complete picture of that, but I'm pretty positive that you can. So for example, canaries, which have tonal sounds, are going to be operating in this region. And beasts like the zebra finch, which generates these very harmonically rich songs, are going to be living in this region in phase space. I would love to do a systematic study with different species, seeing where they are covering a unique bifurcation diagram. And I think it would be very interesting to do phylogenetically comparisons with that. But let me show you a little bit about something I want to share with you, which to me is an open problem, which is the following. Let me show you what happens with canaries. And this is interesting from the point of view of theoretical neuroscience. So this is a problem which I'm very interested in, and to me is still open. And here I'm recording the sound, the frequency, the pressure, and the activity of Vs. Let me focus a little bit on the earth-side pressures. This is a very complicated time series data, if you want. What is surprising me, and every time that I look at this data, is the following. How complex do you think that the brain should be in order to generate this time series data? Because it's kind of sophisticated. And to me, it is really surprising that you can generate all these gestures, but just periodically forcing a structure that has one excitatory population of neurons coupled to one inhibitory population of neurons. So if you take just the Wilson-Cowan oscillator that someone was mentioning today, the Wilson-Cowan oscillator, where you have the description of the rate of spiking of this population, the rate of spiking in here, and you write two differential equations for x and y. The nonlinearity is the sigmoidal function. I'm talking about a very simple dynamical system. X dot equal to minus x plus the sigmoidal of rho x plus the input, which is the sum of x and y through some coefficients, and the same for y. And you drive it periodically, then you can reproduce exactly the same shapes that you see in the pressures of the canary. Now, the interesting thing is that you don't have to fit independently the rate of production with the shape. Once that you decide to drive this thing at a given rate to reproduce the syllable that you have in here, you reproduce exactly the same shape. So something at the level of the neural organization of the brain of this guy, and I don't know where it is, and I don't claim that it's either in HVC, in RA, or where, I have no idea, but just the very low dimensional set of two populations, periodically driven, can generate all the complexity that you have in the earth sac pressure of the canary. That to me is very suggestive. And of course, from time to time, I play with trying to write, given the architecture that we have, plausible ways of getting these dynamics. And of course you can. There are many different ways you can generate this. You can generate them by thinking that it's all generated in HVC or in array. Well, it opens another line of questions, which I'm not going to be talking about today, but I think it's a very suggestive and inspiring thing that such a complicated system at some level can be reproduced by a low-dimensional dynamical system. Say that again? Yes. Yes, for the entirety. Right, exactly. So, for example, you are driving it at some frequency to generate what I call the period one. Then I have another forcing, and I get the other kinds of things. So during the whole simulation, the only parameter that I change is the frequency of the driving. You change the frequency of the driving, and you keep all the other parameters constant. But when you change the frequency of the driving, what you recover is the right shape of the pressure pattern. Yeah. So, let me go then a little deeper into the problem of the pressure, into the problem of the respiration of the birds. And this is something that to me as a physicist was very, very interesting. So there is a little question which seems very silly to me, but I think it was very deep, and was the following. Let's think about not the singing, but let's think about just the respiration. At which rate would a bird breathe? So at which do they produce the syllables during singing, and why? That means, why does a bird sing at the rate that it does, or why does it breathe at the rate that it does? So the behavior emerges from the interaction of a nervous system and a biomechanical periphery. So therefore, I was interested in what is the biomechanics of the respiration. So let me show you how these birds breathe, because this is really amazing. Very different than the way in which we mammals breathe. They have air sacs here, and a lung that is very fixed. So what the birds do is the following. When they increase the volume of this thoracic capacity, on one hand, air goes to the posterior air sacs, and the air that is in the lungs goes to the anterior sacs. And then when they compress the sacs, the air that is in the posterior sacs goes to the lungs, and the air that is in the lungs goes to and the air that is in the anterior sacs goes outside. In this way, you manage to have air, pure air, going in only one direction. It's a very efficient way of having oxygen circulating in your system. So, to complete the circulation of air throughout the respiratory system, you have to go through two cycles, okay? And in the GIFs, I'm showing you the paths of air, and the brown, the blue, they correspond to the two paths of air. So let's see a little model for this. And my little model can be described in terms of a very low-dimensional system again. I'm going to have a variable that describes the motion of the thoracic box that is containing the whole thing. You have the aerodynamic valves that are allowing you to go only one direction. So you are forcing the system, and basically you are interested in trying to compute which is the pressure. Well, you write the biomechanical equations for these dynamics I was describing you before. You write the thermodynamics to describe which is the airflow as a function of the pressure, okay? And we have to take in mind that the bird, when it's singing, can control actively this valve. So when it's breathing normally, these two valves have the same value. So here the flow would be a linear function, and the whole thing will be linear and very simple. It becomes complicated when you try to close a little bit this exit valve in order to sink. And there you have a nonlinear system. So in normal respiration, what you have is a very simple linear system. And here comes another interesting of data science, if you want. How are we going to fit these parameters? Well, from the data, from a data of a bird that is behaving. So if you do that, and here I'm measuring the air sac pressure in normal respiration, and I'm measuring the thoracic movement with just a little magnet and a Hall transducer. I'm measuring that displacement. What I get is a projection of the phase space. I fit the parameters in here, and this linear model has resonances. Where do they have the resonances? At the rate at which the birds breathe. That means at which rate do birds breathe? At the resonance of their biomechanics. So it is true that they have a very complicated neural network in order to decide expiration and inspiration. But they are adjusting to the biomechanics they have to handle. They won't have any normal rate, an arbitrary normal rate, depending on neuroscience alone. You have to study neuroscience together with biomechanics, because you're going to be driving the system at the rate at which you can get the most out of your effort. So at which rate do you breathe? At the resonance of the biomechanics. So here what I'm showing are the resonances of the model that I fitted, okay? And what you have in red is the mean resonant frequency. And in here, the mean respiratory rate for each of the three birds. So they are all breathing at the resonance of the biomechanics. Interesting enough, in previous works, many years ago, when people tried to study the resonances of a respiratory system, they get a dead bird into a pressure chamber, and they did some typical physics experiments, and needless to say, they found a wrong value because that was dead matter, okay? So fitting parameters of a behaving animals allow you to recover parameters that are completely different than if you want to treat living matter like if it wasn't living matter, okay? That's the moral of the tale. Now, because when the bird is breathing, you get data. Yeah, so you have, every bird has different sizes, they have different muscle contexture, so every animal is different. So they will be breathing at different rates because they are of different sizes, basically. One will have more fat, the other one will have less fat, and so on. So, singing, do they sing at any rate? And that is something that I'm really happy about. Now, when you have your model, you have nonlinearities, because now when you are breathing, you have one size in your valves, but when you are doing your expiration, you have to close a little bit the valve in order to produce the sound. So now your model is nonlinear, and now the resonances of this system changes. So, of course, the first thing we did is to check that the model was okay. This is work that a student called Facundo Feinstein is doing. So he measures the activity of the expiratory muscles. He plugs that data into the model and tries to recover the pressure, and it does a very good job. So now he studies this nonlinear system and studies the resonances. And what you get is the following. This is the resonance of the normal respiration, and this is the distribution of the respiratory rates, and this is the distribution of the frequencies of this bird that have a mean at around 14 hertz. Now, this is the resonant curve of the system that is nonlinear. When you have this nonlinearity, the resonances grow, And also they get kind of wild. So probably the bird is also singing at resonances, and this is at least what happens with canaries, and I think it's something that will be general. So birds do breathe and do sing at resonances of their biomechanics. And to me, it was a very interesting lesson that in biology, and in every science, it's always very tempting to be very reductionist and try to study some aspect in particular. But in biology, it's particularly dangerous to do so. You can study the neuroscience of generating respiratory rhythms, but you have to study it hand in hand with the biomechanics, or you're going to be missing big part of the picture, at least that is my opinion. These are things that have to be studied together. So today it has been about the biomechanics of this problem, and the moral of this last part is that the rate at which birds breathe is the one where you have the resonance of the biomechanics. So behavior does emerge. I've been repeating this since I started, but now I believe it more than ever. Behavior emerges from the interaction of the nervous system, a biomechanical periphery, and the environment. Okay, and particularly I think that without studying the nonlinearities in the problem, you won't figure out the biomechanics either. I mean, the problem of where are the resonances of this singing problem, you cannot understand it without understanding that your oscillator is a nonlinear oscillator, and it's a nonlinear oscillator because you are trying to close the valves when you're doing the expiration. And now we're finishing with a complete picture of the biomechanics. And I think we're ready for the third lecture that is going to be trying to use that to understand something interesting about the brain. So instead of going from the brain to the periphery, I'm doing the physicist approach of going from the periphery and use it as a microscope to try to go and study something about the neuroscience. And thank you for your patience.
Learning to sing at those resonant.
Can you ask it again? Sorry.
Yeah. So in a young bird, so young birds, the respiratory patterns are not so well coordinated with vocalization, right? And in part, is it the nervous system learning to sing at these resonances, or is there something else also at play?
So the answer is I don't know, and I think it's a great question. I don't know for many reasons. One, because I never worked with it. I never worked with juveniles. I have a problem with canaries and juveniles that when you work with canaries and juveniles, you have to work at a particular time of the year. And that particular time of the year is where the grad students have vacations in Argentina. So that is one of the challenges. Now, we could overcome this challenge, but it's a very interesting thing because actually it's so interesting because, for example, one thing that one could do to start with is to make a prediction about where the resonance will be because the juvenile has a completely different muscle structure and so on. So to begin with, he has to handle not only that he's learning to do this stuff, but also has a body that is different than the adult. So that would be, to me, a very first question. And then how that body reacts to these instructions that are more fuzzy, that's very interesting. So all I can say is that it's very interesting. And if we think hard enough, I think we can make predictions, but I wouldn't do it in public. Yeah, it's a beautiful question, yeah.
Not changing. It could also just be creating a mental map of trying out different pressures and seeing what kind of vocalizations, like sort of building a map of your biomechanical model. So even if it's a static, like I don't know how much the muscle volume changes during the growth of the canary, but even if it didn't, there might be some mapping happening between the respiratory control and what kind of sound is produced.
Right, right. Yeah, no, it's very interesting because one interesting thing is that if you look at the details of this, when you master, for example, these dynamics between the pressure and the actual forcing, there is a delay. So I think I have the data in this transparency, but if you look at the EMG of the expiratory masses and you compare with the masses, you have some delay. So when you have all this confusion and these instructions that are not periodic, probably there are going to be many mismatches that we could try to figure out and understand. I agree, because here is when you are an adult and you control it, you generate things at which this time difference, for example, don't matter. So, yeah. No, it's a very interesting question. Yeah.
The next slide that you showed. Comparison between the data and the model.
Yes.
Can you show the model?
Yes. So here's the model.
How many parameters did you have to fit to get that in the match?
Yeah. So this f of t is what we measure. And the parameter that we change is f0. And then the only parameter that we change is alpha 0. I would say the ratio between alpha 0 and alpha in. And we use only one value for all the different sea levels. In principle, I would feel allowed to change it if we had to, but we found that it was not necessary. Because what this measures is how larger it is, the valve, when you are closing it than when you are doing the inspiration. So when you are doing the inspiration, it's open. And when you want to do the expiratory gesture, you close it a little bit so that the labia are going to be closer together and start to oscillate. So this is smaller than this. And you change the ratio of these two things. These parameters, the other parameters, are fixed with the simulations of the normal respiration. So you use the values from this model that is only the respiration, and I use them for the regime, for that simulation with expiratory gesture. So these and these are fixed from normal respiration, and this I choose it in order to have the right fitting. And then all I do is to plug the EMG that we measure from expiratory muscles. And what I do not measure is inspiratory muscles, because it's a little bit harder and the dynamics of inspirations is very easy. So what I do is I complete the inspiration with a simple inverted sinusoidal function. But the expiratory gestures are generated by integrating this model.
The question I had was the neural network model that you showed.
Yes.
You can basically create a phase space that one's into in the loaded latent space, but there are no equations associated with it. It's just the time series data in that space that you can reproduce. Right?
Yes.
Whereas all the other cases are actually equations coming from, I guess, what you called, what do you call it?
Biomechanics?
Or
So
there is a difference between the modeling philosophy.
right, exactly.
Is there a way to use, so to say, neural networks on these models and then afterwards interpret what you get?
Right. So let me elaborate on this. You're completely right. So one part of the modeling I feel very confident about, which is the biophysics. So in that I feel pretty confident. With the neural models, no. It's just a phenomenological observation. We are puzzled with Anna since a long time that this complicated behavior is not just a random combination of weights in the neural network that when you do an upsweep give you the right shapes. If you have something that has a plausible neural substrate that is very simple, you get all these specific shapes. So for us, it's just an observation. But it's very difficult now to test it because I mean, the philosophy in the field is going to try to measure unique neurons and stuff, and no, this is some global average dynamics, so it's phenomenological. So if I was a theoretical neuroscientist, or at least if I was a good theoretical neuroscientist, or if I knew about theoretical neuroscience, I would be really tempted to get the information that exists about this neural structure and try to do simulations, conjecturing connectivities, wishing that the average dynamics at the output of the system, which is the activity of N12 and the respiratory activity, PAM and RAM, are the right ones. But it's very blind to me. I mean, how to do that? We don't know. And where can you have these very low dimensional structures whose forcing gives you this? In many places. They could be all in HVC. Or HVC could be just a constant activity because you have a wave of activity and it's in the excitatory and inhibitory populations of RA. What do I know? I don't know. We don't have enough measurements at the right scale to answer this question. My conjecture is that in order to do the measurements at this scale, you have to use lower impedance electrodes and look at global activity. Is not what people are doing in the field. So this part of the modeling is just a curiosity to me, and I'm not actively working on it because there are many alternative possibilities and I don't have clear experiments that I could use to validate one or the other. Patience, my jet lag is catching up and I'm hardly finding the words in English, so thanks for the patience.